Hello and welcome everybody. Thank you for registering for the Endless Form Seminar Series. It's hosted by the Cambridge University Biological Anthropology Society. We're an independent student-led society dedicated to origins research. My name is Jared Schiffert. I'm president of the Cam BioAnth SOC, and I'm here today with our co-host Liam McLean, who's president of the Archaeological Field Club. We offer a warm welcome to our guest speaker today, Dr. Briana Pobiner. Dr. Pobiner is a paleoanthropologist from the Smithsonian National Museum of Natural History and an expert on the evolution of the human diet.
Yes, and Dr. Pobiner has done fieldwork in Kenya and Tanzania, as well as South Africa and Indonesia. And her groundbreaking research has been funded by Fulbright, the Leakey Foundation, and National Geographic, just to name a few. Recent topics which he has covered include human cannibalism and chimpanzee carnivory. And she has recently been part of a team that published a paper questioning the primacy of carnivory in shaping our evolutionary history. And we'll now hand over the time to Dr. Pobiner, and we kindly ask that all questions and comments be saved for the Q&A session at the end. Thank you.
Well, thank you for inviting me. And I'm actually going to touch on that recent paper in this talk. So that was a really nice setup. Yeah, I'm delighted to be here. And I will, first I will share my screen. There we go. Okay. And then I hit this. And then I do that. And now you should just be seeing my slides again, right? Yes, excellent. Okay. So what I want to talk about tonight is the role of scavenging in hominin dietary evolution. So kind of not a broad overview of the evolution of hominin diets, but specifically about the role of scavenging and how we might be able to investigate that. And hold on, I got to move my Zoom controls out of the way so they're not. Okay. There. So I would argue that there have been three significant evolutionary changes in human diets. The first one is the one that I'm particularly interested in and how scavenging might play a role in that. And that's eating food from big animals. And we see the origin of that starting somewhere between 2.9 and 3.4 million years ago. The second is cooking food. And I think we see good evidence for that back to about 1 million years ago, maybe a little bit younger. And the last one is the domestication of edible plants and animals. And interestingly, looks like the earliest domesticates of plants and animals actually aren't edible. There are gourds for carrying and dogs for companionship potentially. But domesticating edible plants and animals, which goes back at least about 12,000 years ago. So what makes our carnivory, basically hominin carnivory, unique compared to that of other primates? So at some point in our history, hominins started to slice meat from bones, bash open bones to get it marrow. One of the things that makes our carnivore unique compared to other primates is tool use and the necessity of using tools in order to consume animal tissues. Our teeth are not particularly well adapted to get into particularly the skin of a lot of the animals that we see butchered in the fossil record. And there right now is a kind of a general coincidence, at least in the timing of the origin of tool technology and the origin of the evidence for butchery behavior in the past. Also eating meat from big animals, certainly primates, like 35 species of, let me step back. So chimpanzees, who are our closest living relatives, they also eat meat from about 35 species of prey. But they tend to be much smaller than chimps. And as soon as we see evidence for meat eating and animal tissue consumption in the archaeological record, it's from big animals. We also do something called deferred consumption. And by that, I mean, humans are weird. We don't walk into a grocery store, get a bunch of packages, sit down on the floor, open them and eat our food. But all other animals basically do that. They consume food as soon as they encounter it. So deferring consumption, not eating food right away, particularly with animal tissue, with meat and marrow, and transporting it to specific places to eat later to share with other individuals, potentially, maybe members of our own family, maybe complete strangers. That's something that's relatively unique compared with other primates. And one thing I would argue potentially is scavenging. There are very few examples of chimpanzees scavenging, eating already dead animals. They don't tend to look at dead animals and go, food. There's a handful of observations. They're very rare. Orangutans and gorillas have never been observed to scavenge. So other great apes don't seem to have scavenging in their repertoire as far as meat eating. So if I was going to give you a whole menu of evidence in general for like the evolution of human diets and even for meat eating, I would talk about chimpanzees and human foragers, and I'm happy to answer questions. Neither of those are my real focus area of research, but I think they can tell us a lot about potentially the origin of meat eating in our evolutionary history. What I will focus on tonight, though, is the Zooarchaeological Record and Actualistic studies. But we can also look at hominin fossils themselves, the archaeological record, even things like residues of blood proteins on the edges of stone tools in the form of ancient molecules, and plant microfossils, things like phytoliths and starch grains stuck on the, basically calculus of hominin teeth. But I'm going to focus on zooarchaeology and actualistic studies specifically as it relates to the potential role of scavenging in our evolutionary history. So a lot of the interest in this topic really started with the hunting scavenging debate. So questions about the timing, the frequency, the resource yield, and the behavioral and biological implications of hominins acquiring animal foods have been, just trying to get my cursor somewhere, okay, have been a part of this hunting and scavenging debate in the scholarly literature for decades. And really beginning several years ago, correlates of hominin hunting were invoked to explain everything from the origins of bipedalism to the origins of technology to adaptations to open grasslands. This is the first of two books that I think were really pivotal for this. This book, Man the Hunter, Richard Lee and Irvin DeVore published this book in 1968. It was based on papers presented in a 1966 symposium on research done on foraging peoples of the world that outlined some ethnographic and some archaeological evidence for the importance of hunting in our evolutionary history. And then not long after that, I should say, this idea also found support in studies of apes, particularly with chimpanzees, with Jane Goodall's observations of chimpanzee hunting published in 1971. But I would argue that the idea of the primacy of hunting in our evolutionary history really goes back further than this, as well as the zooarchaeological evidence. It really has deep roots with Raymond Dart's proposal of the osteodontocoratic, the bone-tooth horn culture, which he proposed in the late 1940s and 1950s. And he proposed this to explain breakage patterns on fossil animal bones in South Africa, found at a few different fossil sites with the remains of Australopithecus africanus, but there were no remains of stone tools. And so he thought that Australopithecus africanus were breaking bones to use as weapons. And this was his osteodontocoratic culture proposal. This actually even made it into sort of the popular literature with Robert Ardrey, who was an American playwright and science writer. He visited South Africa in 1955. He became really enamored with this idea. And it led to what was outlined as the killer ape hypothesis in his 1961 book, African Genesis. And it really outlined the idea for hominins being bloodthirsty, gruesome, savage, maybe cannibalistic hunters. And this was a best-selling book at the time. So that's something thinking about, like how this permeated popular culture. So were hominins hunting from day one of the origin of stone tool technology? So I would argue probably not. This is the earliest solid evidence for hunting technology. And these are spear points from the site of Katupan in South Africa. And those tips have what are called diagnostic impact fractures on them. So based on experimental evidence, definitely used for hunting. And those are dated to about 500,000 years ago. It's very possible that hunting was occurring with archaeologically invisible tools, sharpened sticks, unmodified rocks being thrown at animals, but we really don't have a technological record of hunting technology. We certainly have stone tools going back to, older than 3 million years ago, but not hunting technology until about 500,000 years ago, until maybe we find a better way to detect what's currently archaeologically invisible. So this debate shifted with 2 books that were published both in 1981. The first was Bob Brain's The Hunters or the Hunted. And he used comparisons of modern carnivore den assemblages to reinterpret those breakage patterns that Raymond Dart interpreted as the evidence only for human behavior as the result of carnivore activity. And then also in 1981, Louis Binford published Bones, Ancient Men and Modern Myths, and he presented detailed taphonomic data on bones resulting from prey butchery and consumption from both Nunavut foragers of Alaska, as well as data from wolf dens. And he compared and contrasted these data to look at patterns of bone breakage and bone surface modifications from human and non-human predators. So what does the Zooarchaeological Record tell us about potential scavenging? One of the main traces of butchery behavior are cut marks. And they were recognized, cut marks were recognized on early Stone Age archaeological sites beginning in 1981. And cut marks, I should say, are produced by the contact of sharp-edged stone tools on bones. They were recognized on early Stone Age assemblages in 1981, with a couple of dozen fossil animals from Olduvai Gorge and in Tanzania and Kubifora in Kenya, recognized by Henry Bunn, Rick Potts, and Pat Shipman. A little bit later on is a second trace, and these are percussion marks. And percussion marks are produced during the process of breaking bones open with a hammerstone to access marrow. So cut marks generally meat, percussion marks generally getting marrow. And these were recognized in 1988, also on fossils from Olduvai Gorge and by Rob Blumenschein and Marie Salvaggio. And these, there's even from the beginning of recognizing these in the Zooarchaeological Record, there was a lot of experimentation that was done to kind of try to help us interpret the record based on these marks. And initially, cut marks were always interpreted as indicating access to fully fleshed carcasses. But subsequent research showed that cut marks, or at least there was a proposal that cut marks might be the result of removal of just flesh scraps left over from carnivore consumption. So beginning in the 1980s, several researchers conducted actualistic studies with the aim of producing basically referential frameworks to determine whether the frequency and location of 3 different kinds of marks, cut marks, percussion marks, and carnivore tooth marks, could reproduce varying access to carcasses by hominins and carnivores. And really the aim is to see, can we tell who got there first and who ate what? And generally these referential frameworks focus on limb bone midshafts. They survive a variety of taphonomic processes better than other parts of bones, and they're generally used when calculating bone surface modification frequencies. So this is summing up a whole bunch of that zooarchaeological research. In a hominin-only scenario, when hominins break open bones to get at marrow and butcher bones to get at the meat and cut meat off of bones, you have about somewhere between 5 and 40% of those midshafts preserve cut marks, somewhere between about 10 and 35% preserve percussion marks. In a different scenario, hammerstone to carnivore, so hominins bashing open bones to get the marrow, carnivores coming in afterwards, your percussion mark frequency on those midshafts drops lower to less than 20%. And in a carnivore to hominin scenario, basically a scavenging scenario, where felids get there first in particular, your cut mark frequencies are quite low. usually less than 10%. One problem with these models outlined, though, is that there are several actualistic studies that have found inconsistent relationships between the amount of flesh on bones prior to butchery or the amount removed during butchery and the resulting cut mark frequencies. This is probably because of a variety of basically variables used during these experimental butcheries: the size and species of the carcass, the butchery intensity, what butchery behaviors or activities are being carried out, skinning, disarticulation, and defleshing, even stone tool raw material. And I think cut mark anatomical location may be more important than the frequency of marks in really trying to figure out the order of access of hominins and carnivores to carcasses. So sort of generally, what do we know about shifts in carnivory and where scavenging may have come in? So I think some of the debate in this sort of under this hunting and scavenging framework may be due to the conflation of two important shifts in our dietary history, at least from an evolutionary perspective. One is the initial appearance of the consumption of animal tissues from larger animals, and then is when that consumption became more regular. So really pre-2 million years ago, we see what I would call occasional hominin carnivory. We have two bones from Dakika in Ethiopia dated to 3.4 million years ago that have claimed to be cut marks. There's a lot of debate about whether those are good cut marks. A couple of weeks ago, I think 2 weeks ago, there was a new site called Nyayanga in Western Kenya that was published that has butchery marks on hippos as well as bovids from about 2.9 million years ago. I think there were 12 total bones that had butchery marks on them. But interestingly, they saw two different events of hippo butchery. And those fossils are dated to about 2.9 million years ago. Oops, sorry. And then we see evidence from two other sites in Ethiopia, Gona and Bori, dating to about 2.5, 2.6 million years ago. But really, the entirety of the pre-2 million year old fossil record is 35 bones with cut marks, 10 bones with percussion marks. There's only, if we want to really look at scavenging in the fossil record, potentially, presumably, we might be looking for percussion marks, particularly if carnivores have eaten a lot of the flesh left on carcasses and only the marrow is left. We might expect to find actually a predominance of percussion marks. This was also an idea proposed by Jessica Thompson and colleagues in a paper where they described the hominin predatory pattern. And the idea that actually it's the fat in marrow that may be what hominins are going after. But we actually see very few percussion marks in the earliest part of the Zooarchaeological record. There's only 10 bones out of those, I think 45 total. And some of those actually just may be percussion fractured and not have percussion marks on them. I think some of this may be because percussion marks are a little bit more difficult to identify confidently than cut marks. They may also not preserve as well. So, you know, I think there's still a little bit of like questions about this early pre-2 million year old Zooarchaeological record. At 2 million years ago, there is evidence from the site of Canberra South, which I think does signal a shift towards the beginning of more regular consumption of edible tissues from large animals. And so this is research I've been involved with, not the excavations, but the analysis of the fauna. And there's clear evidence at Canberra from hundreds of butchery marked fossil bones that hominins were acquiring and processing numerous carcasses, relatively complete small carcasses for meat and marrow, and potentially parts of larger carcasses. I think probably still scavenging that larger size prey. They seem to be getting early access to the smaller prey. And the interesting thing about Canberra is that we see this behavior happening repeatedly. It's not just a sort of single occurrence or one level at a site. There are 3 different levels at Canberra where these butchery mark bones come from that span up to about 1,000 years in time. So hominins are bringing fossils to a, central place to butcher multiple dozens of individuals over time. And so this gives us a sense that at least in this time and place, this dietary behavior is potentially becoming more important, or at least more commonplace. After about 1.8 to 1.5 million years ago, we see another shift where I think we have good evidence for potentially hominins having early access to large carcasses. That doesn't necessarily mean they're hunting them, but it means they're able to get there first, or at least control some of the meatier parts of these carcasses. These two photos are butchery marked fossils from Kubifora in northern Kenya. That was part of my dissertation research. And I studied 3 sites that had hundreds of butchery marked bones, but also, again, only a handful, about 27 percussion marked bones versus 272 cut marked bones. Lots of different butchery behaviors going on here, some of which, again, imply early access. There's scraping periosteum to prepare for marrow extraction, but there's a lot of slicing marks on midshafts of bones. And interestingly, at these sites at Kubifora 1.5 million years ago, there is a significant correlation between the frequency of limb elements and the marrow wet weight at one of the sites, and the other site had a nearly significant correlation. So maybe they're transporting or processing limb bones according to marrow yields as opposed to meat yields. Occasionally, so the image on the right is of a tooth mark that is on top of a cut mark. That's like a wonderful smoking gun of hominins got there first, carnivores got there later. So the debate has really continued in the last decade or two, focused particularly on the 1.84-ish million-year-old FLK22 Zinjanthropus assemblage from bed 1 at Olduvai Gorge. On one side of the continuum, Binford originally proposed that the taphonomic patterns observable on the Zinj fauna indicated that hominins were obligate marginal scavengers. They only got flesh scraps and marrow left over from carnivore kills, and that this played a minor role in creating the Zinj site. On the other side, Henry Bunn and Ellen Kroll and others suggested that the same taphonomic patterns indicated that hominins were competent hunters of at least smaller size prey. They aggressively scavenged larger prey. They interrupted feeding predators, and they took what was left of their meals. And they were the major bone accumulators of this fauna. Rob Blumenschein weighed in later in the early 1990s, using some of those referential models to try to determine the order of access of hominins and carnivores, so bringing carnivore tooth marks into the discussion. And then some of Blumenschein's students, Sal Capaldo and Marie Salvaggio, were also looking at applying those referential models to FLK22. And then other research started to weigh in, Manuel Dominguez Rodrigo, and zooarchaeologists really still continue to disagree about how to interpret the evidence from FLK22. And the most recent reanalysis of this fauna was published by Jennifer Parkinson. So I would say overall, though, there's agreement that hominins and carnivores extracted nutrients from carcasses at FLK22 at the Zinj site. And that it's kind of undebatable evidence based on both butchery marks, percussion marks, so cut and percussion marks, as well as carnivore tooth marks and carnivore gnawing damage, and that hominins access both meat and marrow there, again, based on cut marks and percussion marks. But there's still disagreement about whether hominins access largely fleshed or defleshed carcasses at the site. So, but is Homo erectus really that carnivorous? Is there this big shift 2 million years ago that we see starting with Canberra South? And so really these earlier archaeological sites preserving evidence of carnivory, as I talked about, predate the appearance of Homo erectus at about 2 million years ago, but these larger well-preserved sites only appear afterwards. And I was involved in an analysis that quantitatively synthesized data from across sites in Eastern Africa to really test the hypothesis that there was a big increase in the amount of meat and marrow consumption with the evolution of Homo erectus that stayed consistent. And this was the paper. So I've told you the punchline already. There's no sustained increase in zooarchaeological evidence for carnivory after the appearance of Homo erectus. So I'll walk through this a little bit though. So we conducted a broad scale analysis that transcended individual sites. And of course, the individual site analyses are really important. But we really wanted to do this on a regional scale. So we looked at all of the published zooarchaeological evidence dating 1.2 million years ago and older. And you can see the general regions in figure A, the individual sites in figure B, and the hominins potentially that were around at the time in the different types of technology in figure C. And so we looked at these 9 major research areas. Those are in figure A, and the size of the circles that you see are basically the amount of, sorry, where did I go? The size of the circles is proportional to the square root of the number of modified bones reported from each of these zooarchaeological assemblages. So what did we find? So this is the raw time series of paleontological sampling intensity and zooarchaeological evidence for hominin carnivory. And we're putting these in bins of 100,000-year duration. We looked at letter A, the count of total modified bones. Letter B, the count of zooarchaeological sites. Letter C is the count of separately published stratigraphic levels, because sometimes, particularly more recently, there are excavations and publications of individual stratigraphic levels at some of these sites. D is the time series of paleontological species richness, and E is the time series count of paleontological assemblages. There are 2 dotted vertical lines, one at 2.6 and one at 1.9 million years ago. And the pre-Oldowan, so before 2 million years ago, the pre-Oldowan interval, that's all the blue diamonds on the left, before 2.6 million years ago. That has really robust paleontological sampling. Unfortunately, it's excluded from our analyses because there's one zooarchaeological site from this time period, Dakika, with disputed specimens. So we were really interested in the 2.6 to 1.9 million year interval. That's represented by the yellow circles, and 1.9 and later. So the 2.6 to 1.9 million year interval, you can see in letter D, has pretty poor paleontological sampling, and it has low frequency evidence for hominin carnivory in 6 out of the 700,000 year time bins. The period from 1.9 to 1.2 million years ago shows robust paleontological sampling and a high frequency of evidence for hominin carnivory. So we found that the evidence for carnivory really mirrors paleontological sampling intensity. This is the residual evidence of carnivory plotted through time, and it's the same schematic as in the earlier one. So 2.6 to 1.9 is in yellow, 1.9 to 1.2 is in red. And the residual evidence of carnivory reflects the residuals from the regression of each carnivory proxy against the number of paleontological species in that time bin. And that's basically a proxy for richness. The points that fall near that dashed line at 0 have the expected amount of carnivory within a time bin as predicted by paleontological sampling effort. The points above or below have more or less evidence for carnivory than expected based on the paleontological sampling effort. So we don't see a big jump at that dotted line at that 1.9 million years. We don't see a sustained increase really after the evolution of Homo erectus. And over this whole time period, the time bins with more thorough paleontological sampling have more butchery marks, basically. And so this is, pretty much similar evidence. So we found that the site size doesn't increase through time. And so let me just jump back here. So we wanted to see, like, maybe it's just site size increases through time, and that maybe that was a driving factor. And we found, so we regressed the number of modified bones against the number of sites, that's in letter A, against the published levels in all of the sites, and the number of modified bones in all of the sites. And we do see some positive residuals at 1.85 and 1.35 million year time bins, those places where you have much higher than expected butchery for your paleontological sampling. But basically, these seem to be the result of sites with multiple stratigraphic levels. And so again, really in any measure of site size, we don't see a sustained increase in the zooarchaeological evidence for carnivory. So I think we have to couple both the individual site analyses with these more broad scale analyses to really get a good picture of what's happening through time, basically. So Zooarchaeological Record, I think, has pretty good evidence for scavenging, although it's still curious to me that we don't have a lot of evidence for percussion really early on. So I want to turn now to my second main type of evidence, and that is actualistic studies of scavenging. So, if hominins weren't hunting the prey animals they butchered, which it doesn't seem like they were, they may have obtained edible carcass resources. That's anything edible from a carcass: muscles, brain, viscera, marrow, skin, bone grease, by potentially two main modes of scavenging that have been proposed. The first is passive scavenging. That's waiting until the primary consumer is completely finished consuming their prey and going in and getting whatever's left. The assumption with passive scavenging that you would have a marginal amount and quality of animal tissue. The second type of scavenging that has been proposed is confrontational scavenging. That is chasing carnivores or the primary predator off of their kill before they're done, and interrupting them and getting what's left over. And there is an assumption that there would be an ample amount of tissues and a variety of animal tissues from active scavenging or confrontational scavenging. And what have previous studies told us about passive scavenging? So let's start with that. So it's been assumed to involve opportunistic, infrequent access to nutritionally marginal animal foods that may be pathogenic, may be putrefied. But I think modern observations counter this assumption. So there are two main varieties. So I talked about passive and active or confrontational or aggressive scavenging, but two main varieties of passive scavenging have also been hypothesized. The first is this sort of scavenging from temporarily abandoned kills of solitary predators, such as tree-stored leopard kills between feeding bouts, which you see pictured here. So again, sorry, this is the assumption of what you would get from passive scavenging. There's only been one pilot study of the persistence of tree-stored leopard kills. This was done in the Serengeti area of Tanzania by John Cavallo and Rob Blumenschein. And they found that these partially eaten kills were always abandoned for prolonged periods of time when the leopards went off and came back and ate and went off and came back and ate. They persisted as scavengeable food resources for an average of 28.3 hours. Compared to terrestrial kills, they found that the tree-stored kills were more predictably located, that they were less seasonal than terrestrial kills. They required no pursuit effort, low search effort, but the potential animal tissue yields from tree-stored leopard kills have not been really extensively investigated or quantified. So I think there's more work to be done there. I do think this pilot study is really intriguing though. But the main passive scavenging niche that has been proposed for hominins is permanently abandoned kills of social or solitary terrestrial felids, particularly lions or saber-toothed cats. And this is predicted due to either satiation of the cats, of the felids, they are finished and leave a lot left over, or just anatomical limitation to extracting the remaining food. And so this is what is proposed to be this passive scavenging niche. small amounts of abandoned flesh, and all marrow from abandoned felid kills, lions, saber-tooths of medium-sized adult herbivores. And as I mentioned, felids are generally thought to be the optimal predator to scavenge from. They have more limited ability to access within bone resources than hyenids or canids. And they often don't consume all the flesh on larger carcasses. So Rob Blumenschein outlined a few variables that could affect the quality of this kind of passive scavenging opportunity: the size of the carcass habitat, the initial consumer type who got there first, and the ecosystem predator-prey ratio. This can change seasonally, where there are migratory herds of herbivores, and this really affects the competition level for carcasses. Maybe we can detect some of these in the archaeological record. So there have been several previous studies of passive scavenging, particularly in East African ecosystems. So in the mid-1980s, Rob Blumenschein did a passive scavenging study in the Serengeti and Ngorongoro ecosystems of Tanzania. He found that the carcass availability was highest in the dry season and in riparian woodland habitats. In the mid-1990s, Martha Tappan did a scavenging type study. It wasn't actually the central focus of her research, but she did her research in the Virunga National Park in Zaire, which is a wet savanna ecosystem. She found that this had year-round carcass availability, particularly in open floodplains versus riparian forests. Manuel Dominguez Rodrigo did a scavenging study in the late 1990s, and he worked in the Masai Mara in Kenya, which is basically the same ecosystem as the Serengeti and Ngorongoro. It just happens to be over the border in Kenya. He found that carcasses in riparian woodlands hardly had any scraps of flesh at all, that there were only flesh scraps in the open, I should say plains, not plans, only flesh scraps were only available in the open plains, and they were not available in woodlands. So interestingly, different than Blumenschein's findings. And then in the late 2000s, Agnes Gidna did a study in Tarangire National Park in Tanzania in a savanna woodland environment. And she found that most carcasses consumed by lions in the open plains were defleshed, and that flesh scraps were only available in bushy plains, I said plains again, plains, and gallery forest, but not in open plains. And so there's not really a lot of consistency in previous studies as far as where scavenging opportunities, kind of the smaller parts of habitats that we might expect to find them. So I did a passive scavenging study in the early 2000s in a different kind of ecosystem than where most of these studies had been done before. I spent 7 months in a private game reserve in Kenya, now called Olpejeta Conservancy. You can see where it is on the map of Kenya, where it's zoomed in, where that red star is. And I simulated passive scavenging. And I particularly targeted Olpejeta because it was a lion-dominated, and still is really a lion-dominated ecosystem with few spotted hyenas. And most models of hominin scavenging are based on interactions with large felids like lions or leopards or saber-toothed cats. So I thought this was actually quite a good model for a felid-dominated prehistoric ecosystem. So my study methods, I would find out about a carnivore kill from a variety of sources. I would wait until the lions were completely satiated. I was not particularly interested in simulating aggressive or confrontational scavenging. And then, these two lions in this top right photo, these were two young males that hunted together a lot. I suspect they were brothers. The one on the left has a giant belly full of warthog. He's not going anywhere. So I'm just, sitting and watching as they moved off of their kill. And then I would go in and document what was left. And I actually then also would, after I did that documentation, I collected the carcasses, put them in the back of my 4-wheel drive vehicle. And I had an assistant with me cleaning off all the bones so that I could also study the chewing damage left by different predators on those bones. Separate questions, but, still sort of part of the same study. So here's my study sample, and I'll go through the data collection. So I collected 24 kill samples from several different species of carnivores. I want to focus on the lion sample. That's my largest sample, and I really think it's the most relevant for simulating scavenging. And I divided the prey into two size divisions that are really related to how we classify prey in the archaeological record. Larger prey are larger than 250 pounds. So things like zebra, particularly zebra were the dominant herbivore, still are at Olpegida, and larger antelopes like eland or buffalo. Smaller ones are less than 250 pounds, things like impala and gazelle and warthog. So I documented the location of where meat was left and estimated the amount of meat left after the lions had their fill. And I really divided things into bulk flesh and flesh scraps based on previous definitions from other scavenging studies. Flesh scraps are defined as less than 10% of the original flesh left, including small packets of flesh still adhering to the bone. This was the definition that I used. It was published approximately less than the size of a normal human's palm in area, but larger than 2 to 3 centimeters and 150 grams. So like something worth eating, I guess. That's what flesh scraps are. So what did I find? How much meat can a scavenger eat from those larger animals like zebra? I'm going to walk through the axes before I show you the data. On the x-axis are basically different parts of carcasses based on limb bones that are grouped. Hind limb to the left, then forelimb, then torso, and then the head. And on the y-axis is the percent of those bones that have different levels of flesh availability, coded like a traffic light. No flesh is red, flesh scraps are yellow, and bulk flesh is green. So from large prey, lots of green and yellow. About 50% of bones were abandoned with large muscle masses still on them. Those are in green. And when you combine them with bones abandoned with flesh scraps, 95% of those larger kills had something to eat on them, basically. And what about small prey? Same axes, very different data. Almost no bulk flesh left on smaller prey. About half of the bones had flesh scraps. So a big take-home study from a big take-home message from my scavenging study is that the size of prey makes a big difference. So how do we translate this into something more quantifiable? We can use the weights of an adult wildebeest pictured here. And this is a common size of animal found in the archaeological record. So using an estimate of 4 calories per gram of meat. We'll get to that in a moment. So a fleshed hind limb of a wildebeest weighs about 19 pounds. A defleshed hind limb, about 10% of the flesh left, just scraps, weighs about 2 pounds. A defleshed carcass would potentially have about 12 pounds of meat on it. The estimate of 4 calories per gram of meat, you get about 2,200 calories from a wildebeest that only has flesh scraps left on it. How does that translate into something a little bit more understandable? Almost 24 McDonald's burger patties worth of meat on a defleshed wildebeest carcass. What did this mean for Homo erectus, let's say? There has been an estimate that Homo erectus daily caloric requirements were somewhere between about 2,100 and 2,300 calories a day. So potentially an adult wildebeest, a defleshed adult wildebeest with only flesh scraps on it, could feed a Homo erectus for a day. That's without even breaking the bones open, the limb bones open, to get at the marrow inside. A non-fat depleted adult wildebeest, not seasonally stressed, you can get about 3,000 calories of out of marrow, out of those bones. Less than 30 minutes of processing time, your next door neighbor Homo erectus gets to eat as well. And the net yields of extracting brain and flesh can be even higher, sometimes 2 orders of magnitude higher. So I think we should think about reframing this debate. I think at the root of this debate are important questions about the evolutionary consequences of the changes in hominin behavior and ecology with this dietary shift to acquiring, consuming, and sharing animal tissues. But here are some questions that I think would be interesting to think about in this debate. Is there a functional association between stone tools and fossil bones, particularly butchered ones found in the archaeological record. What animal, when and who basically was doing this consumption? Maybe that's harder to answer in the fossil record. What is the likelihood that the earliest animal tissue consumption included scavenging? I think it's pretty high. What animals were being eaten, in what landscape settings, in what habitats, in what seasons? How often did consumption of animal tissues occur? How much did animal tissue consumption contribute to the diet relative to other foods? When did carnivory become important or a common part of hominin diets? How predictable was the availability and acquisition of animal foods by hominins? And did basically eating animal tissues lead to food sharing? These are important questions that I think can reframe this debate in a way that is more productive. So I think what we can do for next steps in asking these questions, we always need to examine more fossils, to go back to archaeological sites where they may not have been studied, looking for zooarchaeological evidence of hominin carnivory as in butchery marks. We can develop better taphonomic models as well as better taphonomic methods in order to, I think, better identify order of access to carcasses. I think observing more small felid kills and even some natural death assemblages to kind of widen the referential frameworks for understanding scavenging is another important next step. And I think conducting experiments on carcass tissue edibility persistence. And by that, I mean, how long can a carcass persist and those tissues be edible before they're just not basically safe to eat at all? So that's it. I'm stopping there. I want to acknowledge particularly for the scavenging study that I outlined, research permission, funding for a variety of the research that I've done, and support from folks at the National Museums of Kenya, Ol Pejeta Conservancy, and some of my mentors.
Great. Well, thank you so much for that. And I'm glad that you included a list of questions at the end, because that was kind of a big question I had about sharing. You know, what are the chances of sharing, especially if the cost is so high in terms of, you know, if you have to chase something off or maybe.
I'm gonna stop
go.
sharing my screen and my slideshow and stop sharing my screen. Let me get back over here. There we go. Okay.
We'll open it up to questions now. And if you want, you can either unmute yourself and just ask, or you can message either Liam or I. You can raise your hands.
I guess oh, okay. Zavdi?
Yeah, it's Zodi. Thanks very much for the great talk, Briana. I was just wondering what you think of it's the Nyanga hippo that they found, what you think about that, because it's a very interesting discovery with these two Paranthropus teeth. So I'd be very interested to hear your thoughts.
Yeah, no, it is really interesting. And it's funny because, so I do research in Kenya. This team does research in Kenya. They've been excavating this site since 2015. So I've just been waiting and waiting. I'm sure they've been waiting with more excitement for it to be published. You know, I'm not really surprised that they have found another evidence of, like, you know, one or two individuals being butchered. I think it's pretty cool, again, that it's large animals, that there's more than one hippo individual. There's also bovids that are butchered. And I was kind of amused to see how much the press ran with, and they found Paranthropus teeth there. I'm going, They also found Paranthropus in the 1950s at Olduvai Gorge in the same level with butchered bones. So this is not new. They're, you know, Paranthropus and early Homo, whether it's Homo habilis or Homo erectus, have been found at least kind of in the same general vicinity. At FLK 22, there's both Paranthropus and Homo found there. So, you know, I don't think it necessarily means anything about who's doing the butchering, but I do appreciate it opening up more the idea that maybe we have to let go of the idea that it was only Homo that was making and using stone tools for butchery. So, but I think it's great. It's great evidence.
And I know it's not your area of expertise, but there were these cut marks, right? Were you sort of convinced that, sort of, unlike the speculation surrounding Dakika, that these could confidently be attributed to hominin activity?
Yeah, no, and actually cut marks definitely are a strong area of expertise for me, and I was convinced by their cut marks. So I think it's good, solid evidence. Yeah.
Great, thank you. It's very interesting. Yeah, and I found it really fascinating how much meat you can get from a carcass, right? It's
Well,
really
hippos are massive, and so yeah. And it's interesting because I think, is like I said, as soon as we see the evidence, not as much from Dakika, but from Bori, from Gona, from Nyanga, we're seeing butchery of large animals. So, also at Olduvai and other places, there's butchery of small animals, but I think it's interesting that there's butchery of giant extinct elephants, of hippos, of giraffes, of, other really large animals.
great. Thank you very much.
I'm happy to go next, Briana. Thank you very much for the presentation. It was really, really interesting. And since we are on the topic of large animals, my question relates to something that you also said, how prey size is something that is really important in terms of how carnivores or hominids may interact with the carcass.
Mm-hmm.
And something that you are very aware as well is that dealing with a large carcass using stone tools can also do different patterns of cut mark quantities, percussion mark quantities.
Mm-hmm.
So having those two things in consideration, I was wondering to what extent may prey size be a relevant factor that should have been accounted for more in the bar paper.
Yeah,
perhaps if we are seeing larger
Yep.
animals, we see less cut marks, but a lot more meat consumption.
That's a really good question. And I think, I feel like that paper was a really, hopefully useful and important compilation of data. I think given the supplementary material, it's like, yeah, a next step could definitely be to go in and say, like, were these large or small prey? Sometimes it's not reported, so, depending on what, where the data are coming from. But I do think it would be interesting to see what the patterns are like on different size prey. I think that would be a really interesting next step.
Especially since we have places like BK in Olduvai where really large animals are quite predominant.
Exactly. And I do think there's, like you said, like prey size makes a big difference. And there's also questions about with really large prey, how much, how many butchery marks will you expect anyway? You can, all the proboscidean people, you can butcher a proboscidean and not leave a single mark. And so that can potentially complicate your expectations of butchery mark frequencies. So.
We agree on that.
Oh, and I guess I was wondering, you brought up the example of the wildebeest deep flesh carcass having like enough calories, like 2,200, to feed a Homo erectus for a day. But I was wondering how likely you think it is that Homo erectus actually would have survived like that, because I was thinking that's enough for one Homo erectus to just get by for one day, assuming they can get every morsel off. And you found like 24 carcasses across 7 months. So
right. It's a really good question. And I think also, probably something I should add on that last slide of other, research we need to do is the expectation of carcass encounter rate. I've seen some of like different expectations published, and what's the likelihood that a Homo erectus you know, assuming you would like plop one down in today's ecosystems, what's the likelihood of them encountering one carcass a month, one carcass a week, you know, and what state might that carcass be in? And no, and I should, you know, I would never expect that like Homo erectus or any hominins going around subsisting solely on like defleshed carcasses. But I think thinking about it as, in a sense, like a resource that is beginning to be more often incorporated into hominin diets. You know, I don't know how we would be able to, and I have to think more about this, like how could we estimate encounter rate? How could we estimate like changing frequencies of encounter rate in different habitats, in different, like with different seasonality? So yeah, it's a good question.
Yeah, indeed.
Certainly, I think, you know, using data from modern forages, I always like to go, oh, like, I don't want to just sort of impose modern foragers as models for what Homo erectus would have been like, but would have been like. But I do think, you know, data from the Hadza and other modern foragers certainly have a place in thinking about, you know, estimating things like encounter rate and things like that.
Mm-hmm.
Well, thank you so much, Dr. Pobiner. Is there any other questions from the chat or from anyone else? Well, if not, first, I'd like to thank Dr. Pobiner for taking the time to talk to us today, especially now that she's doing collections work in the Czech Republic. And thank you, Liam, for co-hosting and bringing the AFC into this. And thank you, everyone, for joining. We encourage you to check out our website and check out the AFC website for more fascinating seminars by experts like Dr. Pobiner, and to look out for the upcoming seminars. Well, thanks so much.
Thanks for having me. Thanks for
Thank
the
you
good
very much.
questions too.
It's really interesting.
Thank you. Yeah, thanks for your questions.